Hello, my name is Claire McLeod, and today I will be talking about the Briggs-Rauscher reaction, which is an oscillating reaction. It's a very unique reaction, and it's similar to the iodine clock reaction that we did during class, in the sense that it changes color due to the reaction mechanism and the reaction rate. So what is the Briggs-Rauscher reaction, or even an oscillating reaction? Oscillating reactions are ones that can be visually classified due to the color change that happens cyclically. On a basic level, an oscillating reaction involves a complex mix of chemical compounds, and this complex mix can be seen and demonstrated through an equally complex reaction mechanism, which usually involves a plethora of steps, although most often those steps can be condensed into two of the more important or two of the most significant overall steps. These steps lead to periodic changes in the concentration of a certain compound, and so within the reaction mechanism, while one step may increase the concentration of a single compound and lead to a color change due to that increase in concentration, the next step could simultaneously decrease that concentration of the compound and lead to a very sudden color change, which is something you see, but that eventually subsides due to the impact of limiting reagents. So today, the two most connected topics that I will be talking about in relation to my demonstration are reaction mechanisms and reaction rates, two of my favorite chemistry topics. But before we get into the reaction itself, we have to prepare 3 solutions that we will then mix to create this demonstration. Our first solution, solution A, is simply 100 milliliters of 3% H2O2. Solution B is made by dissolving 7.25 grams of potassium iodate in 100 milliliters of water and adding 4.3 milliliters of 6 molar H2SO4. And finally, solution C is made by dissolving 0.55 grams of manganese 2 sulfate and 2.6 grams of malonic acid in 100 milliliters of water with additional starch to enable us to see the color change. To begin this demonstration, I've added 50 milliliters of solution B, which was the potassium iodate solution, and 50 milliliters of solution C, which was the manganese 2 sulfate malonic acid starch solution. To this, we also added 100 milliliters of hydrogen peroxide, or solution A, which is what initially caused that first color change to amber. Eventually, as you can see, the reaction goes on and the amber instantaneously changes to the dark blue color, before the dark blue color slowly shifts to a colorless and then back to an amber solution. This color change, as I've talked about, occurs to a multitude of steps and complex compounds, which we are about to talk about now. So what exactly is going on with this color change in this demonstration? As you saw, upon the addition of hydrogen peroxide to solution B and solution C, the reaction vessel appeared amber. Then, almost instantaneously for the first cycle, it turned to a dark blue, before that same dark blue shifted to a colorless solution, and then that colorless solution eventually returned to amber. This cycle of color changes is due to the 2 different processes that are happening within that reaction vessel. So here is a line to split up our 2 steps. The first step, which is what's changing the amber to the dark blue, is a fast reaction. This fast reaction has a high reaction rate, which means it is producing a high amount of HIO, or hypoiodous acid, very quickly. And this quick production of hypoiodous acid is due to the the iodine in the solution reacting to form HIO. This, because the iodine in the solution is reacting, it has a low concentration of iodide, which leads to the dark blue color, because iodide in solution is amber. This part of the reaction is also a radical reaction, which isn't something that we have talked about in class, but it is a reaction that involves highly reactive species. And due to the high reactivity of those species, the resulting reaction is fast. So on the other side of this reaction, we see that it in some way has to be changing the solution from dark blue to colorless to amber. And to do this, it is somewhat of an opposing reaction to the reaction I've just explained on the right. On the left, we have a slow reaction with a low reaction rate. So that means that the reaction doesn't produce HIO quickly or in high amounts. In fact, this side of the reaction, or this step of the reaction mechanism, leads to an increase in the concentration of iodide, which is what yields that amber color. This process is non-radical, which means it involves species that do not react readily or quickly. And that is why we see on this side of the reaction that the color in the vessel changes from dark blue to colorless and then to amber versus instantaneously from amber to dark blue. Now that we know somewhat of an overview of the two processes that are taking place on either side of the reaction, let's put some chemical equations to those processes, because I know how much we all love our chemical equations. So step number 1, we have iodate, the IO3-1, reacting with H2O2, or hydrogen peroxide, and the excess acid from H2SO4. This yields hypoiodous acid, or HOI, excess oxygen, and excess water, which we don't care about when it comes to color change. So on the left, as a reactant, you see the iodate, which is yellow or amber in nature due to the presence of iodine. And then on the right, as a product, we have hypoiodous acid, which turns a dark blue when it complexes with the starch. So this first step is what you'll see that it's the step that's changing the solution from amber to dark blue. And that, as I previously explained, that happens almost instantaneously when the buildup of hypoiodous acid immediately complexes with the starch to produce that color change. In reaction 2, we are going to see how the dark blue solution filled with hypoiodous acid yields and returns back to that amber solution that contains iodide. So as a reactant in this step, we have hypoiodous acid, which was the product of step 1, and that is the dark blue solution. In this step, it reacts with malonic acid from our solution C, which is the CH2, CO2, H2. When this reaction takes place, it yields a complex compound made up of iodide ions and malonic acid, which you can see in this big chunky compound as a product. This iodide ion turns the color back to an amber due to the presence of iodine. And as we learned before, this is the reaction that is slower, and so we also see that colorless interface between the color change. So as we finish up this reaction, I want to make it clear that this reaction does stop approximately after 10 minutes when the hydrogen peroxide, or iodate ion, has been used up, and it limits the reaction, and because that iodate has been used up, the reaction ends as the dark blue solution.
I'm a scientist. I'm a true scientist. Woo!